Hello everyone, I'm Professor Cong. Welcome to the class of Advance of Infection and Immunity. I'm from Qilu College of Medicine, Shandong University. This class, we are going to talk about coronavirus vaccines, COVID-19 and current health crisis, the severe acute respiratory syndrome coronavirus SARS-CoV-2 causes coronavirus disease 2019, also called it COVID-19, emerged in late 2019 and spread globally. Recent dramatic increase globally, especially in the United States. By the end of 2020, there are over 20 million reported cases, about 2 million deaths. Treatments are being identified but have limitations. The only way to return to normal lives may be with safe and efficacious vaccines. So far, more than 90 vaccines are being developed against SARS-CoV-2 by research teams in companies and universities across the world. Researchers are trialing different technologies, some of which haven't been used in a licensed vaccine before. First, let's introduce the importance of SARS-CoV-2 spike protein. Vaccine development is focused on the principal target of the humoral immune response, the spike S glycoprotein, which mediates cell entry and membrane fusion. The SARS-CoV-2 S gene encodes 22 unlinked glycan sequences per promoter, which likely play a role in protein folding and immune evasion. So, how coronavirus infection induce the host adaptive immunity? Coronavirus SARS-CoV-2 enters the body. The virus surface spike protein lock onto ACE2 receptor on the surface of human cells. Virus enters a cell, virus fuse with vesicle and its RNA is released. The virus RNA translated into proteins and virus release. Virus ingested by antigen-presenting cell, we call it APC cell, and activate T helper cells. T helper cells enable B cells make antibodies that can block the virus from infecting cells, as well as mark the virus for destruction. Cytotoxic T cells identify and destroy virus-infected cells. So, the long-lived memory B and T cells that recognize the virus can patrol the body for months or years, providing immunity. So, the top priorities for vaccine development. COVID-19 vaccine development has followed normal vaccine development principles. First, is the vaccine must be proven effective, meaning it can help prevent COVID-19 in at least a majority of vaccinated people. Second, the vaccine must be proven safe, with robust safety data generated from thousands of people. Third, the vaccine must be consistently manufactured at the highest quality standards. In response to global health crisis, we progressed our program safety while ensuring highest compliance and quality standards and ensuring safety. So, how many vaccine types are developed now? So far, there are virus vaccine, viral vector vaccine, nucleic acid vaccine, and protein-based vaccine. From the figures, we could see the numbers of vaccines in development. First is the virus vaccine. Virus vaccine has weakened virus and inactivate virus. So what is weakened virus? A virus is conventionally weakened for a vaccine by being passed through animal or human cells until it picks up mutations that make it less able to cause disease. We can thus SARS-CoV-2 by altering its genetic code so that viral proteins are produced less efficiently. The second virus vaccine is inactivated virus. In these vaccines, the virus is rendered uninfectious using chemicals such as formaldehyde or heat. Making them, however, requires starting with large quantities of infectious virus. The second type is viral vector vaccines. There are around 25 groups are working on viral vector vaccines. A virus such as measles or adenovirus is genetically engineered so that it can produce coronavirus proteins in the body. These viruses are weakened so they cannot cause disease. There are two types of viral vector vaccines. They are replicating viral vector and non-replicating viral vector. What is replicating viral vector? This is the newly approved Ebola vaccine. Is an example of a viral vector vaccine that replicates within cells. Such vaccines tend to be safe and promote a strong immune response. Existing immunity to the vector could blunt the vaccine's effectiveness. However, such as weakened measles. The second non-replicating viral vector, such as adenovirus. No licensed vaccines use this method, but they have a long history in gene thereby. Booster shots can be needed to induce long-lasting immunity. Next is protein-based vaccines. Many researchers wanted to inject coronavirus proteins directly into the body. Fragments of proteins or protein shells that mimic the coronavirus outer coat can also be used. They have protein subunits and virus-like particles. The protein subunits vaccine. There are 28 teams are working on vaccines with viral protein subunits. Most of them are focusing on the virus spike protein or a key part of it called the receptor binding domain. Similar vaccines against the SARS virus protected monkeys against infection but haven't been tested in people. To work, these vaccines might require adjuvant, immune-stimulating molecules delivered alongside the vaccine, as well as multiple doses. Next is virus-like particles. Empty virus shells mimic the coronavirus structure but aren't infectious because they lack genetic material. 5 teams are working on virus-like particles VLP vaccines, which can trigger a strong immune response but can be difficult to manufacture. Next is a nucleic acid vaccine. At least 20 teams are aiming to use genetic instructions in the form of DNA or RNA for a coronavirus protein that promotes an immune response. The nucleic acid is inserted into human cells, which then throw out copies of the virus protein. Most of these vaccines encode the virus spike protein. RNA and DNA-based vaccines are safe and easy to develop. BNT162 vaccine is the vaccine that is getting to be used on people. The mode of action of the BNT162 vaccine candidates are as follows. Spike mRNA formulated in LNP enters cell. mRNA is released and spike protein is made and processed. APC present S protein fragments which submit to CD4 helper T cells, activates T and B cells. CD8 cytotoxic T cell eliminates virus infected cells, potentially increase length of protection. B cell produce virus neutralizing antibodies, bind spike proteins, and prevent virus infection of human cells. Memory T and B cells provide immune memory to ensure longer-term protection against SARS-CoV-2. So the advantage of mRNA vaccine. First is the safety. They are non-infection, chemically defined, no viral foreign proteins. Second, the efficiency. The mRNA vaccine is broad immune response, minimum risk of anti-vector immunity, and permits frequently boosting. Third is rapid response. mRNA vaccine, the technology enables rapid development and quick production scaling. So far, there are several vaccines will begin to be used. I think we will return to normal life very soon. Vaccines will protect us from coronavirus finally. Okay, this is the end of our lessons. I hope from the study you could get to know more advanced in infection and immunity, and could develop more vaccines to help people against the infectious pathogen. Thank you for your listening.